Hi everyone, in this PowerPoint I want to explain a method that is commonly used to map a gene to a chromosome. This method is called somatic cell hybridization, and for many of you, you may find mapping papers that use this technique. So hopefully this short PowerPoint will be helpful in explaining how it works. So here we have some example data where we're screening 3 different hybrid cell lines. You can see they're numbered 23, 34, 41. Those numbers are just arbitrary. And then in each row, it tells you which human chromosomes are present, as indicated with the little picture of a chromosome. So in the cell line number 23, human chromosomes 1, 2, 3, 4 are present, but 5, 6, 7, 8 are not. And in that cell line, they've assayed also for 4 different gene products, A, B, C, and D. And in this cell line, we can see that there's a minus sign for genes A and C, but a plus sign for genes B and D. Okay. So that's how to understand how these tables are set up. And the first question one might ask is, what chromosome could gene A be on? So we can think about this as follows. We're going to use the information in all 3 of the cell lines to answer this question. We can eliminate chromosomes 1, 2, 3, and 4 because in cell line number 23, those chromosomes are present, but the gene product A is absent. Right? That's the opposite of a correlation. In cell line 34, we see that gene A is expressed and chromosomes 5 and 6 are present. We've already excluded chromosomes 1 and 2. The other chromosomes present there are 5 and 6, so perhaps gene A is located on chromosome 5 or 6. We then look to see what other information we have from the 3rd cell line, number 41. Gene A is expressed, and in terms of chromosomes 5 and 6 that we've narrowed it down to, 6 is missing, but 5 is present. Therefore, gene A correlates perfectly only with chromosome 5. So we would map gene A to chromosome 5. I'm going to pause here just for a minute and suggest that you try to solve this kind of problem for genes B, C, and D using the information on this slide. And then in just a minute or a half a minute, I'm going to go ahead and animate and show you the answers. So you can pause at this time to see if you can get the correct answers. Okay, so here are the correct answers. For gene B, we would probably map gene B to chromosome 3. That's the chromosome that correlates with the presence of gene product B. Gene C is not expressed in any of those cell lines. And chromosome 8 is not present in any of the cell lines. But that's a negative correlation, so that's why I wrote chromosome 8 question mark. What we would really want to see is try to get another cell line where chromosome 8 is present and hopefully gene product C is expressed. And then lastly, gene D, we would probably assign to chromosome 1 because it is present in all 3 cell lines, both chromosome 1 and gene product D. Okay, so lastly, I just want to show you what data from a paper might look like. This is the McGinnis et al. 1984 paper, and in this paper they were mapping a mouse gene, the mouse 10 gene, to a chromosome. So on the left, this is a southern blot, and they took DNA from 10 different mouse hamster cell lines. So their hybrids were using mouse cells and hamster cells. And then they ran the DNA from those 10 different cell lines in each of the lanes and probed for the presence of the mouse 10 gene. And so when we see those positive bands, first of all, I guess I should explain, on the far left are the controls. So we have mouse DNA where we see a strong band and hamster DNA where we do not. Okay. So we have a positive control and a negative control on the left of this blot, and then we look at those 10 cell lines, and we see bands in 4 of the 10 cell lines that I've put a bracket at the top that are positive for the presence of mouse 10. Okay, so that's just how you actually figure out if your gene of interest is present or not. That's one way you can do it, is by doing a southern blot, and then you know whether or not that gene is present in those cell lines. Okay, so most of the time, authors will actually just summarize results from somatic cell hybridization experiments. So the table on the far right is more commonly seen, and this is summarizing the results from southern blots of 35 different cell lines. Okay, so the southern blot we just looked at was just a representative blot for 10 of those 35 cell lines. Okay, within the table, the leftmost column shows the mouse chromosome number, and then the number of cell lines that were concordant, which means they had both or neither that chromosome and the mouse 10 gene. And then it also goes on to tell you the number of cell lines that were discordant, which means that the presence of that chromosome and the mouse 10 gene did not match up. Okay, so if we kind of quickly scan through, what we're really looking for are zeros in the discordant columns. And so there's a little arrow that I've put a blue box around. For mouse chromosome 6, you see the arrow on the far right. It's pointing at the zeros, a total of no discordancies. That means there's no failed correlations. And for every other chromosome, there is at least one discordancy, which means, right, a failure to match up the presence or absence of the mouse chromosome and the mouse 10 gene. So the mouse 10 gene was mapped to chromosome 6 in this particular study. Okay, so where to find mapping experiments in OMIM? Okay, and this is going to be important for section 4 of your research paper. What I'm showing you here is an example for Apert syndrome, and what you would do is click on the link for the gene associated with the disease. And then, so in this case, for Apert syndrome, the FGFR2 gene is the gene that's associated. So after clicking on FGFR2, you would see the table of contents as shown here. So we've got in blue on the left below the different sections. And if we click on mapping, which I've circled in red, you would see the paragraph that is shown here on the slide. And what you can see where I've underlined in red is that the Dion et al. 1992 paper actually was able to map this gene to chromosome 10 using human rodent somatic cell hybrids. Okay, so they didn't say it was somatic cell hybridization, but we see words that are very close to that. They're using somatic cell hybrids, and they mapped the gene to a particular chromosome. So you could find that paper and see their actual data, and you could actually explain that experiment as a key mapping experiment for this gene in this disease. So that was just one example. Another thing to note is that not just for this particular example, but in others as well, you sometimes will see that the name of the gene and the protein don't match. Here they're calling it the Beck gene, B-E-K, and that does encode the FGFR2 protein. Okay, so sometimes that can be confusing, but if you run into any issues or you get stuck, please feel free to email as always, or check in during office hours or during our regular class times. Thanks.